Thank you for the introduction, Alex. And there's sort of this tradition that you start out your seminar by thanking the organizers for inviting you. And I think this would be a bit odd in this situation. So I'd like to start out by thanking Joan for catching COVID. So I had an excuse to put myself on the schedule. So what I promised in the title to talk about was phosphorylation inside disordered signaling complexes. But that's actually just a dirty trick, because I would like to start talking about something completely different. So I would like to invite you to take part in a thought experiment. And the question I want you to consider is the following. What is my risk of being eaten by a tiger today? So a few of you may be thinking that this is an extraordinarily stupid research question. And you're right. And the reason that you're right is because the concentration of tigers in Denmark is exceedingly small. It's not quite zero, but it's very, very small. And being eaten by a tiger is similar to what we in physical biochemistry would call a bimolecular reaction. So bimolecular reactions require that the components meet before anything can happen, and thus they are concentration dependent. Because the concentration of tigers is so low, regardless of how good a predator a tiger is, then my risk of being eaten by a tiger is effectively zero. So now we're going to introduce a single perturbation into this thought experiment. So we keep the concentration of tigers in Denmark constant, but now I'm holding one of them on a leash. So what does that do to my risk of being eaten by a tiger? So I think we can probably all agree that the risk goes up by many, many orders of magnitude. And this is, again, something we can explain using physical chemistry, because now, since the reactants are linked, this is no longer a bimolecular reaction. So it's a unimolecular reaction. So we don't need the molecules to find each other in the solution before they can react. So the velocity of the reaction, or the reaction rate, is independent of the concentration. So this resembles something we often find in biochemistry. If you imagine that instead of the tiger, we have an the interesting thing here is that it's not, since it's not determined, the contact is not determined by the concentration. Instead, it's determined by the nature of the linker, the nature of the connection between the tiger and the prey. So if you now imagine that the enzyme is the tiger is in fact not a tiger, but instead it's an enzyme, and the person at the other end is its substrate, then this is actually the situation that I would like to talk about. And this is incredibly common in biochemistry. There are many enzymes that are somehow tethered to their substrates. A good example would be the kinases. There are many kinases that have docking domains that don't contribute to catalysis, but serve to target the kinase to the right set of substrates. It's equally important in phosphatases, where you have these little slim binding pockets that, again, do not play a part in the active site, but bind the peptide and brings it close to the active site. So the common features of these enzyme docking interactions are that you need some sort of disordered linker to connect the binding site and the active site, and that the docking motifs are often short linear motifs. So this is something that is well founded in the IDP world. And the reason that I actually ended up working on this, I think, is that this is maybe one of the clearest examples of how the structural ensemble of IDPs regulate biochemistry. So if you have a kinase that's tethered to its substrate, then the probability of them coming together is determined by the linker. So that means that anything that happens in the linker will become some sort of regulation. So I think there will be loads of regulatory events that happen not by, say, changing the activity of the enzyme or the properties of the substrate, but actually changing how they are connected. And we haven't really started to sort of not even to scratch the surface of this yet. So in other words, IDRs, they a lot of the talks in this series has addressed how you get from the sequence to the ensemble, and this is where I started my career as well. But then you get your ensemble, and then you realize that maybe this was actually the easy part. This was the easy part of this pathway. That actually the more difficult part was getting from the ensemble to the function here. This is what I would like to talk about, because linkers are maybe one of the clearest examples of how the conformation ensemble of an IDR directly regulate biochemistry. Good. So the question that we're asking is what sort of quantitative model we should use to describe an enzyme that's tethered to its substrate by a disordered linker. And this is a slightly different question from what we would normally be asking in sort of mechanistic enzymology, because normally the question would be how does the rate of the reaction depend on the on typically the concentration of the substrate, and then we have the Michaelis-Menten equation, and we have many other things that looks a bit like it, but none of these work because they cannot describe intramolecular reaction or intra-complex reaction. So it's not just a question of finding the shape and the magnitude of this curve. We actually also have to start out by figuring out what we want to put on the x-axis here. So somehow the independent variable is the ensemble of a disordered protein. So this is something that's very general, and we think there's a general answer, and the best way to go for a general answer we thought was to build a model system, and a minimalistic model system that allowed us full control over the parameters of the system. So this is what Matti did. So what he did was to take the catalytic domain of protein kinase A and then tethered it to its substrate via disordered linkers, where we can change the length of these linkers, and then we have a coil-coil interaction that we use to stick them together so we can prepare them separately. And we can also change the properties of the substrate. So when you take this system and then measure the rate of phosphorylation, so this is a single turnover reaction. So we have to do it by quench flow. So you can see it happens in the sub-second regime. So when you compare a tethered and an untethered reaction, then as expected, we see a massive boost of the single turnover reaction, up to 500-fold. But then we can repeat the same experiment. We can repeat it with substrates of different qualities, and for each of these we can vary the length of the linker. So this is the same experiment repeated for all these. So what you have on the y-axis here is the single turnover rate of the reaction. So you see that in all cases the rate is slower the longer the linker is. So far so good. This makes a lot of sense. But we can also see that the slope, the magnitude of the dependence on the linker length differs between these substrates. So for the best substrate, we only see a twofold change between the longest linker combination, which is around 200 residues, and the shortest one, which is about 20 residues. Whereas for the worst substrate, we see a tenfold increase in the single turnover rate. So this is what we would like to somehow explain. But again, I said that I wanted to go for something that was sort of a more general description of a tethered catalysis. So then we have a problem with our x-axis here, because so this is the length of a glycine serine linker, and that's not very general. But the trick here is that it allows us to controllably vary the connection, but we need to turn that into something that's modular. So the question is, how do we quantify interactions that are linked, and then by extension, how do we measure it? So the quantity that I will focus on for the rest of this talk is something called the effective concentration, which is very intuitively straightforward to understand. So if you tether a ligand to what it binds to, then the effective concentration is what governs the binding rate, such that you'll have the same encounter rate as you would have with a similar amount of free ligand to the effective concentration. So this provides a number for how closely things are linked by the linker. So how do we measure this? So this is the work that Alex described from Charlotte, summarized on half a slide. So one way you can measure it is by doing competition experiments. So what Charlotte did was to build a fluorescent biosensor where we have a tethered protein interaction where we can change the nature of the linker. Charlotte did more than 200 different linkers, and it's actually not high throughput by physics. It's low throughput by physics, but a lot of it. So it took us a couple of years. With this biosensor, then you can do a titration where you titrate in the free ligand and you thus displace the tethered ligand, and then you get a titration curve that looks like this. And then from the midpoint of that transition, you can quantify the effective concentration. And then you can do this for different linkers of different length, and then you get something that scales like this. So it follows a polymer scaling law. So this is a power law similar to what has been described for end-to-end distances and other measures of sizes of IDRs. So this is actually also what we expected, but we also use this system to study how the linker sequence affected the compactions of the IDRs, but I will not talk about that today. But for most applications, if you have a linker and you want to get some idea of the effective concentration, then this is probably not what you want to do. So this took a couple of years. Instead, you can do something that takes a couple of minutes. So you could use a polymer model, for example, the worm-like chain, where this has already been established a couple of decades ago, that you can describe effective concentrations enforced by linkers pretty well if your IDR can be approximated by a homopolymer model. So the math behind it looks like this. So it's simple. There's just a lot of numbers, and I have actually not managed to type down this equation without making some mistake. So it's not very practical. So what we decided to do instead was to make a web applet, and this is a collaboration with Julie and Lucia from Buenos Aires that you can visit. It's called CF Calculator, and then you can plot in your parameters for how long your linker is, what kind of persistence length you have, what kind of distance between binding sites, and then you can get a pretty reasonable estimate of what a disordered homopolymer linker would give you in terms of effective concentration. So that's actually how we normally start. But actually, in this is relevant to this audience where you have a lot of people working with structural ensembles, then the effective concentration should, in principle, also be something you could calculate from the structural ensemble, because the effective concentration is essentially built, or it depends on the end-to-end distance distribution and the probability of these coming together at or approaching at a certain distance. So you can take your ensemble, you can calculate the end-to-end distance distribution, and then you can convert that into how the effective concentration of something that was tethered with this sort of IDR would scale at different spacings. Again, the math of this is fairly simple. So basically, you just divide it into thin shells and then divide by the volume, and yeah, it's fairly So it's fairly straightforward. So in principle, this would be a way to functionally interpret ensembles of linkers. So then I've reached the age where I start to care about semantics. So I would also like to just spend 1 minute to talk about the sort of the terminology of linkers. So when we talk about intrinsically disorder or talk about linkers, what we normally think of is this a continuous IDR that links different functional entities. But a linker is also sort of a functional designation. It's something that links things that interact. And the connection, if you look at the connections in signaling complexes between enzymes and the substrates, then they're often much more complex than just a single IDR. So I would like to propose that we invent the term linker architecture to sort of describe this much more complex kind of molecular bridge that can be composed of several different rigid and flexible elements. So to study these more sort of complex IDRs and show that this is relevant, we studied this type of linker architecture. We have a folded domain surrounded by IDRs, and what we can show is that basically you can boil down. So this is a simple passive model without sequence specific contacts between the linker and the domain. And for a model like this, you can boil down the effect of the linker to the end, to the distance between the N and C terminals of the domain. So it acts as a rigid link that has the same length as the attachment point, the distance between the attachment points. And this, we can also actually show that such linkers where you have a folded domain can, when you have to span something, if you have to span a long distance, then having something that's more rigid is actually allows you to have a higher effective concentration, because you can focus the ligand on that distance. So in some cases, it may actually, even though we normally think of linkers as something that's very IDR centric, then you can actually have a better linker if you have something that's more rigid, if you have to span a large distance. Great. So this was all really a really big detour. So if you remember where we left off the kinase, then we had the situation that we had a lot of kinetic data for different linker lengths. That was this unsatisfactory x-axis. So I think you may have gotten the idea now that what we want to do is to take this linker. So actually, it's a linker architecture because we have the folded domain in it, and we want to convert that into a more general x-axis of the effective concentration. So this is basically what we did using a polymer model. So when you take these single turnover rates, and you can replot them. So instead of linker length, then we have the effective concentration that linker length enforces. And then you get a plot like this, and for the other ones, you get something similar that's shifted. So that is something we know and like as at least as enzyme people, because it looks just like the Michaelis-Menten equation, except that this is not steady state kinetics. This is single turnover kinetics, and the x-axis is not substrate concentration, but the effective concentration. But the curve shape is the same. So we can describe single turnover kinetics using a Michaelis-Menten-like dependence on effective concentration. And we would like to propose that this is actually something that is general to all sorts of tethered enzymes. When you can do sort of the theoretical modeling without telling the system that you're working on a kinase, and then you get this behavior out of it. So the effective concentration here serves as this unifying way of quantifying the linker architectures, and this allows us to functionally predict here that when you change the linker to generate a higher effective concentration, for example, by collapsing the linker or bypassing it, then that will not only will it increase the rate of the tethered substrates, it will also change the substrate usage towards weaker substrates. Good. So this was all single turnover kinetics, but only getting a single turnover out of each enzyme is a little bit unsatisfactory. So most enzymes actually work in the steady state regime, where they continuously go through the cycle. So if we go back to our sort of model of the tethered kinase as the tiger tethered to its bait, then what we have to change. We have to change the question such that it's not what is the risk of being eaten, that's the single turnover event, but rather a steady state kinetics question of how many people can a tiger eat per day. So this is what we would like to do, and the way we do it is to make a reversible protein interaction between the substrate and the enzyme. So if we just do sort of a back of an envelope calculation, then we can probably envision two scenarios here. So you can have a very tight binding. So what will happen then is that you will get efficient targeting. The first substrate will be processed, and then nothing will happen, right? Because the enzyme is still tethered to the substrate that it has already processed. You can also go to the other extreme and have a very weak targeting interaction, which would also not work very well, because a very weak targeting interaction is essentially the same as not having a targeting interaction. So we have to find a sweet spot in between these two events, where the binding is not too tight, or it's just not too weak, but it's just right. So what we're looking for is some sort of Goldilocks binding affinity for tethering of enzymes. So we wanted to study this using the same model system that Matti built before, and actually we had a reversible interaction, a reversible connection, but this is very tight. So when you so it's tight enough that the dissociation happens on a much lower time scale than the turnover rate. So we could use it to study single kinetics. But what Matti did then was start mutating the interface, and by a combination of 3 different mutations, we can get this series of different docking interactions, where we can continuously tune the binding affinity by more than 1,000-fold, mainly through the off rate. And then we take these enzymes, and then we go back to the kinetics, this time doing steady state kinetics, and then we ask the question, how does the affinity of the docking interaction affect the steady state turnover? And so this is the work of Nico, who took over the project from Matti, and the data always looks something like this. So just a quick explanation of the x-axis here. So this is off rate. So we have strong binding means a low off rate, and weak binding means a fast off rate. So what we see here is this apex at the middle, as we predicted. So we have this regime over here where the reaction is limited by tight binding, so we get product inhibition, and then we have this other regime with weak interactions, where we have weak binding and poor targeting. So we would like to actually, but we would also like to be able to predict where this Goldilocks zone is. So I think I can think of several good reasons that this will be useful. So first of all, we have a lot of docking motifs out in nature, and sometimes they mutate, and they get mutated and cause disease. So if we have a mutation in a docking interaction that presumably would probably lower the affinity of the docking interaction, then that can have 2 different effects on on the overall throughput of the pathway, depending on which side of the docking of the Goldilocks zone you are on. So we can both increase or decrease the efficiency of the enzyme. Also, we would like to be able to design new enzymes and use tethering systems to improve enzymes. And if you want to do that, then you have to know where your optimum is. So we used our model system to study how the position of the apex depended on the conditions of the system. So first of all, the substrate that we use. Again, so we know that we have this weak binding regime where we don't get very much binding to the weak binders. But again, we figured that we could probably compensate for that by using a higher substrate concentration. On the other hand, we have this tight binding regime where you have that the binding, the length of the binding interaction is much longer than it needs to be to complete a docking cycle or a catalytic cycle. But again, so this will also depend on how long it takes for it to predict or to go through a catalytic cycle. So we also predict that we have poor substrates will need longer time, so they will need stronger docking interactions in order to have an optimum. So first of all, testing the substrate concentration, that's very easy. So you can see what happens here is that, so this is low concentration, then we increase it tenfold, and then the position of the apex shifts to weaker interactions as predicted. Similarly, we can test the quality of the substrate, again, using these point mutations that each reduce the kcat over km by a factor of 10. Kcat over km is a measure for how good a substrate is. So the data that I started out by showing you is actually for the worst substrate. So what happens when we go to the better substrate is that, first of all, we push the position of the apex to the right as predicted, and eventually we end up pushing it outside the range that we can actually measure. And then for the very good substrates, then we basically just measure the same rate as we would without having a tethering interaction. So we would also like to explain this quantitatively. So we try to define a minimum reaction scheme where you can envision two different pathways. So we can either have the normal pathway where the substrate just phosphorylates or the enzyme phosphorylates the substrate without a docking interaction, and then we have a reaction where the docking interaction forms first, and then the matching happens. So that gives us two routes, and then there are also some ternary pathways, but they're not very important unless you have very high concentrations. So defining this reaction scheme allows us to do numerical simulations. So basically, we say these are the rate laws for the system. This is the mechanism. Here are the concentrations. Here are the rate constants. Calculate what the rate will be. And that allows us to test the properties of the system very quickly. And again, you see we get this Goldilocks zone where we reproduce the same behavior, where we shift the position of the apex when we increase the substrate concentration to weaker interactions. Similar to actually to predict it, to be able to put a number on where the apex is, then Matti, he then defined a rate law for this tethered reaction path, ignoring all the other pathways, and it then ends up looking like this. So it looks a little bit complicated. So this is the expression for the rate, but when you then take the derivative to get the maximum, then a lot of the terms disappear, and you end up with this very nice and simple expression, which quite well predicts the apex of the tethered system based on the properties of the substrate and the docking interactions, the effective concentration and the substrate concentration. So this is what you need in order define where the apex is. So so far I've only talked about the position of the apex. You may have noticed as well that there's a huge difference between how much the reaction is enhanced as well. So for the optimal substrate, we saw basically no enhancement, and we see a massive enhancement for the worst substrate. So this then also, I think we also want to explain which conditions will provide the largest enhancement from a tethering interaction. And we can describe this using 5 simple rules for how you can when an enzyme reaction is enhanced most by tethering. So we enhance catalysis most if we have this medium strength Goldilocks docking strength. Also, if the docking interaction is fast on and off, if the linker maximizes the effective concentration, and if the enzyme and substrate is poor, so that gives us more scope for improvement. And finally, if the substrate is present at low concentration. And again, we tested this for kinases, but we think it's a general principle for anything that would follow a similar mechanistic scheme. So I think this takes us to the conclusions. So I think the main conclusion is that intrinsically disordered linkers are massively underrated. I think they are probably the least loved IDRs of all. They're usually just chopped away or ignored compared to stuff that binds to other things. But IDRs directly affect biochemistry in a way that you can actually explain from the ensemble. So I talked about tethered catalysis. This could also be about binding, intermolecular binding. There's a lot of auto-inhibited proteins out there. It could be about the avidity of the multivalent interactions. All these things are also affected by the properties of the disordered linkers. The effective concentration allows us to think quantitatively about linkers and linker architectures, and provides a functional interpretation for structural ensembles. So I think that brings us to the most important slide. So I would like to thank the people from my lab. So the work that I have talked about today was done by 3 fantastic postdocs. So the first 2 postdocs that joined my lab, Matti and Charlotte, and then Nico has taken over the project since. These other people, they work on equally interesting projects that I would be happy to talk about another time. And finally, if you liked this kind of science, then we have an open postdoc position that I think you should apply to. And that was all. So thank you.